Welcome back to the deep dive. Today we have a highly visual, incredibly special deep dive for you. I mean, imagine you're managing a highly critical logistics network, right?
Okay, I'm picturing it.
You're a lab researcher at the bench, a biotech executive looking at the bottom line, or maybe a process engineer trying to optimize a facility. You are absolutely feeling the acute pain of traditional AAV purification bottlenecks.
Oh, absolutely. Adeno-associated virus downstream processing is notorious in gene therapy development. It's the hurdle everyone has to clear, and historically, it's just where so much time and material is lost to, well, complex, highly manual, busy work.
So our mission today is to explore exactly how you can compress weeks of that tedious downstream laboratory work into just a few hours. And because this is a highly visual roadmap, we're going to walk through a fascinating presentation from GeneMedi, step by step, slide by slide, covering their one-day downstream gene therapy solution.
Yeah, and the phrase one day is not just some catchy marketing hook here. We're going to look at the underlying science of a complete structural overhaul and how a daily lab functions.
Let's lay the groundwork and jump right into the visual material. Starting with slides 1 and 2, we get a bird's eye view of the landscape. GeneMedi is presenting a massive portfolio here.
They really are.
They detail innovative solutions spanning across biologics, cell therapy, and gene therapy. It's just an incredibly comprehensive overview of the field.
And understanding the scale of their operations gives important context for the rest of the presentation.
Yeah.
GeneMedi isn't just trying to sell you a single isolated product like some random buffer solution or a standalone filtration column.
Right.
What they are presenting in these first two slides is a complete integrated ecosystem. They're building the infrastructure for novel AAV discovery, development, and industrial manufacturing all under one roof.
That end-to-end mindset brings us directly to the core problem you are likely facing right now. Moving on to slide 3, we hit the ultimate value proposition bypassing those traditional AAV bottlenecks.
The pain points are very real.
The slide perfectly illustrates it. First, you have traditional ultracentrifugation. Anyone who has done this knows the dread, right? Multi-day spin times, highly labor-intensive protocols, and terrible parallel scalability.
It's just brutal for efficiency.
And then you have the industrial alternative AKTA or FPLC systems.
Yeah, and those systems solve scale, but introduce an entirely new set of headaches. FPLC stands for Fast Protein Liquid Chromatography. These are massive, incredibly complex industrial chromatography refrigerators that cost an absolute fortune.
And they aren't exactly plug and play.
Not at all. They require dedicated experts just to program them, and they lock you into sequential processing constraints. You have to process one single sample after another. So there's a massive dependency on high capital expenditure for the equipment alone.
But look at what is positioned right next to those legacy methods on slide 3, the GeneMedi benchtop ecosystem. It promises complete processing times of under 4 hours total. Yeah, and it allows for high parallel processing of dozens of variants simultaneously. And the craziest part, it's either completely equipment-free or totally automated. Let me stop and ask the obvious question here. Are we completely bypassing the massive capital expenditure of buying and maintaining complex FPLC machines just to get high-purity AAV?
For the vast majority of discovery, screening, and benchtop work, the data suggests absolutely yes. By engineering the purification matrix into a benchtop spin column format, you completely eliminate the queue for that expensive FPLC machine.
That makes total sense.
Right. If you have 30 new viral variants to test today, you don't line them up for an FPLC over the next month. You just spin them all at the exact same time in a standard tabletop centrifuge.
That is a fundamental paradigm shift for a lab's budget and project timeline. But how do we actually achieve this without the multi-million-dollar hardware?
Well, the workflow is key.
Now looking at slides 4 and 5, we see the mechanics. They lay out the end-to-end workflow, and it's beautifully simple. It's a 3-step process. Step 1, capture your virus with their PurPro-X AAEZ. Step 2, polish the sample with AAV full. And step 3, quantify the results with the TrueX titration kit.
You really have to pay close attention to the causal flow illustrated on these slides. This isn't some patchwork pipeline where you buy a capture resin from vendor A, spend a day trying to figure out a complex buffer exchange.
Our buffer exchanges are the worst.
They really are. And then passing it to a quantification kit from vendor B. No, this is a continuous, streamlined pipeline. The chemical eluents from one step are specifically designed to flow directly into the next.
And friction between steps is exactly what kills momentum in a lab. So let's zoom in on that very first massive hurdle. Transitioning to slides 6 through 11, we get a deep dive into the capture phase.
The AEEZ spin columns.
Yes, and the speed metrics here are genuinely shocking. They show a complete purification protocol from raw cell lysate to purified AAV in a flat 30 minutes. 30 minutes. You're just using a standard benchtop centrifuge, no ultracentrifuge required.
And the biological engineering enabling that speed is what makes it work. The presentation includes these electron microscopy images highlighting a custom pore structure of approximately 25 nanometers in the resin. In chromatography, pore size completely dictates mass transfer.
And 25 nanometers isn't a random number, is it? An AAV particle itself is roughly 20 to 25 nanometers in diameter. So these pores are basically custom molded to catch these specific viruses like a perfectly sized lock and key.
Precisely. By engineering the pores to that exact dimension, they maximize the internal surface area available for the large AAV viral particles to bind, even when the liquid is flowing through at high speeds.
Which explains the efficiency.
Exactly. That mechanism is how they achieve an exceptional binding capacity, exceeding 3 times 10 to the 14th viral particles per milliliter of resin.
Depending on your specific targets, they offer different variants. There's variant U1, which is their versatile broad-spectrum option binding multiple serotypes like AAV2, 5, 6, 8, DJ, and 7-mele8. But they also offer variant U2, which is explicitly engineered for high-affinity capture of AAV9.
And AAV9 is heavily prominent in modern gene therapy, specifically for targeting the central nervous system and cardiac tissues. So having a dedicated high-affinity resin for it is crucial. Beyond the sheer binding capacity, though, the durability of this matrix is remarkable.
Yeah, the gel electrophoresis data for the U2 column backs that up. They show AAV9 samples maintaining distinct, intact VP1, VP2, and VP3 capsid protein bands. And for those who might be a little rusty on their virology, those are the 3 essential structural proteins that physically snap together like microscopic Lego bricks to form the virus's outer protective shell.
Right, and seeing those intact bands with virtually no host cell protein impurities, even after 5 consecutive cycles of purification and regeneration, proves the resin doesn't degrade. The purification efficacy shows no noticeable decline after repeated uses,
Which stretches a lab's consumables budget significantly. But the real proof of any of this is what happens when you introduce the virus to living cells. They compare the in vitro efficacy of AAEZ against the old standard of traditional ultracentrifugation.
and the flow cytometry analysis reveals a massive disparity. When infecting 293 T cells, the ultracentrifugation method yielded a 14.2% transduction rate. The affinity chromatography via PurPurX nearly doubled that, achieving a 25.2% effective transduction rate.
Why is there such a massive jump? I mean, it comes down to the physical health of the virus, right? Ultracentrifugation subjects the viral particles to immense physical shearing forces for days. It beats the virus up.
It really does.
The benchtop spin columns are gentle and fast, leaving you with a significantly healthier, more potent viral vector that's much more capable of infecting target cells.
And that potency translates directly to living models. The presentation shows in vivo validation using mouse models. They injected the AAEZ purified vector systemically via the tail vein, and the subsequent imaging shows robust systemic luciferase expression.
The mice are basically glowing exactly where they're supposed to glow, proving these vectors maintain their fundamental biological integrity and natural tissue tropism.
Which is the ultimate goal.
Okay, so we've conquered the benchtop. We've proven we can move fast in R&D with highly viable vectors. But if I'm a biotech executive, my brain immediately jumps to the commercialization cliff. What happens when this drug actually works? Moving to slides 12, 13, and 14, we answer that exact question. Benchtop spin columns are great for screening, but what happens when my project needs to scale up to industrial manufacturing?
You certainly can't run a commercial clinical production facility using tabletop centrifuges.
Exactly.
The brilliance of the PurPurX system is that you don't have to change your underlying science. The system outlined here is seamlessly scalable because it relies on the exact same proprietary AAV pure affinity resin.
So it's a one-to-one transition.
Yeah, they offer this identical chemistry in pre-packed columns in 1 milliliter and 5 milliliter sizes for immediate plug-and-play integration into those AKTA or FPLC systems we discussed earlier.
So the multi-million-dollar FPLC isn't obsolete, it's just properly reserved for when you genuinely need commercial scale, rather than bottlenecking your daily discovery work.
Spot on. You do your high-throughput screening on the benchtop to find your winning therapeutic variant. Once you have a winner, you scale that exact same purification chemistry to your FPLC using the pre-packed columns.
And for massive industrial manufacturing.
For that, you can purchase bulk resin bottles ranging all the way from 10 milliliters up to 10 liters. Because you aren't changing your underlying chemistry when you scale, You completely eliminate the massive headache of redeveloping your entire purification process from scratch.
And the recovery rates at that commercial scale are phenomenal. The data shows AAV2 recovering at 90.73%, and AAV5, 6, 8, and 9 are all comfortably above 93%. That is uncompromised vector recovery.
Which is exactly what you want at that scale.
High yield is fantastic, but it means nothing without high purity. On to slides 15, 16, and 17, we tackle the next major headache in gene therapy polishing. Specifically, we have to separate the full capsids, the ones actually carrying our precious genetic payload, from the empty ones.
Right, because empty capsids are a major critical quality attribute for regulatory approval and clinical safety.
Think of it like sending a massive fleet of delivery trucks to a patient's cells, but half the trucks are entirely empty. It triggers all of the body's immune system alarms, causing potentially severe side effects without actually delivering any of the life-saving medicine.
That's a perfect analogy. Separating those empty delivery trucks historically requires incredibly complex ultracentrifugation gradients, or very tricky anion exchange chromatography that often involves tedious, error-prone buffer exchanges.
And nobody likes those.
No. The PurProX AAV Full Enrichment Kit completely bypasses that complexity. It's an instantly accessible benchtop solution that directly processes the elements that just came out of your AAVZ spin column.
But how does it actually tell the difference between a full microscopic shell and an empty one?
It exploits extremely subtle surface charge differences between the two. The negatively charged DNA payload sitting inside a full capsid subtly alters the overall electrical signature of the particle from the inside out.
Oh, that's clever.
Yeah, the proprietary AAV full matrix detects that slight electrical variance. You load your sample directly without an intermediate buffer exchange, wash out the electrically distinct empty capsids, and then elute the full capsids.
The visual proof they provide is striking. They show a separation chromatogram achieving the flat loading phase and distinct elution peaks you'd normally only see on an expensive FPLC, all done via a benchtop matrix. And the functional proof backs it up. They ran a flow cytometric analysis comparing the baseline unpolished mixture to the final full capsid product.
And the results. The unpolished baseline mixture drove an 8.89% transduction efficiency in their test cells. The waste fraction, the empty capsids, showed almost zero activity at 0.24%, confirming it's truly payload deficient.
Makes sense.
But the final product, the enriched full capsids, that transduction rate rocketed up to 42.9%.
That is nearly a 5x increase in baseline transduction efficiency, completely fuss-free, right at the bench. You've captured it fast, you've polished it beautifully, but, you can't manage what you can't measure.
Very true.
Looking at slides 18 and 19, GeneMedi urges us to rethink titration with their TrueX ELISA kits, because legacy PCR methods have some serious fundamental flaws.
Yeah, quantitative PCR has been the industry standard for titration, but it requires incredibly complex multi-step sample preparation just to extract the DNA for counting. It's highly susceptible to amplification bias and false positives.
Which just makes interbatch variability incredibly frustrating when you need precise clinical dosing metrics. Let's use an analogy here. Using PCR titration is like trying to count the number of cars in a massive parking lot by dismantling all the engines, multiplying the spark plugs in a lab, and doing the math to guess how many cars you started with.
That sounds like a nightmare.
It is messy and prone to massive amplification errors. The TrueX ELISA kits rely on direct antibody binding. It's like just walking into that same parking lot and putting a physical tag on the hood of every single intact car. You are directly, clearly counting the actual intact product.
And GeneMedi engineered these direct counting ELISA kits for uncompromising linearity and stability. They offer the TrueX1 Universal Kit, which applies across most standard serotypes, and their UniQ line, providing highly specific serotype detection.
The precision data they show is pretty remarkable, isn't it?
It really is.
Yeah.
The coefficient of variation, or CV, measures the relative variability of a test. For their AAV5 specific kit, the CV is a mere 1.03%.
Let's put that 1.03 per CV in real-world terms. A 1 per CV basically means if you sit there and measure the exact same sample 100 times in a row, the results will be virtually identical every single time. No more second guessing if your titer is artificially off by a factor of 2 because of a weird PCR amplification quirk. You have absolute confidence in your dose.
That confidence is everything when you're moving toward clinical trials.
Exactly. Let's zoom back out and look at what all of this means for a real human working in the lab. Bringing it all together on slides 20 and 21, they summarize this massive paradigm shift. They lay out a timeline comparing the traditional workflow to the GeneMedi workflow, and the contrast is eye-opening.
The timeline really crystallizes the literal one-day downstream advantage. In a traditional setup, you dedicate days 1 through 3 entirely to FPLC programming or ultracentrifugation purification.
Ugh.
Then day 4 is completely eaten up by complex PCR titration prep and analysis.
But with this integrated ecosystem, the entire process is completed on a single day. Hour 1 is capture with AEEZ, Hour 2 is polishing with AAV full, and hours 3 and 4 are quantifying with the TrueX ELISA.
That is a staggering reduction in manual laboratory labor, equipment downtime, and just sheer frustration.
You are generating high-purity, highly enriched capsid AAV ready for immediate downstream biological assays on the exact same day you started.
And when you review the core applications they list, pilot-scale studies, rapid construct screening, academic R&D, the compound impact of this speed becomes obvious.
Oh, absolutely.
If a lab is doing capsid-directed evolution, generating thousands of mutated viral variants to find the optimal therapeutic one, they simply cannot afford to wait 4 days to evaluate each batch. High-throughput screening demands high-throughput purification.
Or consider the rise of small-dose production. If a team is working on localized therapies, say a highly specific injection for ophthalmology or a targeted neurology application, they do not need a 50-liter bioreactor run. They just need high-quality, high-purity vectors incredibly fast without wasting their critical source materials on the massive hold-up volumes inherent to giant FPLC systems.
Right. By removing the friction, this ecosystem empowers the researcher to iterate faster. In biological development, the speed of iteration is literally the speed of discovery.
Which brings us to the next step for your own lab. Finally, on slide 22, GeneMedi provides a direct call to action. If you are watching this deep dive on YouTube, this is where you want to pause the video and grab your phone.
Yeah, get those cameras ready.
GeneMedi has placed QR codes right there on the screen. Scan them right now. You can download the full detailed brochure to share the science with your team. Even better, scan the second code to request a free sample. You can test this one-day downstream transformation in your own lab and see that 30-minute capture speed for yourself.